Assalamu alaikum student. Today we will talk about the second law of Mendel, which is the law of segregation. The word segregation means the separating. So actually in this law, some separating of genes occur. In that time there was no the theory of genes. So Mendel said that it is a law. It is an experiment which he presented through which some picture or separating from each other, like these are the two pictures. Normally we say it's a genes, but he said it is a pictures. So these are two pictures which control a specific character of body, like tallness, like color of the body. So when during the gamete formation, they will separate from each other like this, okay, and this will go this, but this will go to one gamete, this will go to another gamete, but they cannot go to one gametes. They cannot go. So if we talk about the definition, so every individual contains two factors for a single character, and they separate from each other during gamete formation, and each gamete receive one or other but not both. So I said that these every individual contain a pair of genes or a pair of character which control a specific characters in our body. During the gamete formation, they will separate from each other and will go to opposite gametes, okay, but they cannot go to one gametes, okay. So Mendel performed this law of segregation. In previous lecture we studied about the law of dominancy of Mendel, in which the F1 generation were produced, but we only saw about the phenotype of the F1 generation. And when the Mendel performed his experiment, and he performed experiment by the cross pollination, in which he cross pollinated a pure tall plant with a pure short plant. But it wondered the Mendel that what are the reason that all the F1 generation were tall plant. So Mendel said that are they all F1 generation are identical to the tall parents, so they are tall, or they also contain some material, some elements from the short plant, or it is a pure tall plant, pure tall, or it also contain the material of the short plant. It is mixed. So to find this in answer to this question, Mendel performed another experiment on the F1 generation to find what are the nature, what is the genotype nature of the F1 generation. So if we're starting the lab segregation, we will talk about the genotype of the F1 generation, that what were present, what was the genotypic nature of the F1 generation that permit and help the Mendel to apply the law of segregation on the F1 generation, and then form the law of segregation, the separating of genes, and promote the law of segregation for the Mendel, and also prove the talk of the Mendel that it is contains some materials from the short plant. So we talk about this. So Mendel first took one tall plant and second short plant. So I said that every individual has two genes, has two factors. So he took, this is the one, the tall plant contain two factor or two genes, which is, but this is a pure plant, so they contain two tall pectors. The both pectors are tall, and the short both are short. So he cross pollinated tall with tall, the short. So the tall goes to the, you can see here, the tall goes to the short, and then the tall goes to the short, and then short goes to the tall, and short goes to the tall. So the result of this was the tall, capital T, short, small t, capital T, small t, capital T, small t, capital T, small t. So this causes all were tall plants. Plants were tall, and he called these as F1 generation. So if you look at a genotypic nature of F1 generation, which and the phenotypically it is tall, but you look at a genotypically, so you can see there that one is the capital T, which represent the pure tall plant, and second is the small t, which represent the pure small, the short plant. So therefore the F1 generation contain both the tall plant materials and also the short plant materials. So on this way the Mendel then proposed, then presented and applied another experiment which is called as a law of segregation of Mendel. So let's come. Then by performing the law of experiment of the segregation, which then lead to the construction or the performing of the law of segregation, Mendel lit these two F1 generation, which were tall phenotypically, but genotypically it will also contain the short plant. So it let these two self-pollinate. It means it self-pollinate. It is a plant, and it contain in from the male part to the female part. So it contains both tall plant and also short plant, but phenotypically these were tall. So he self-pollinated these. So if you look at it theoretically, so genotypically, so we can say that this tall plant with the tall plant, tall capital T with the tall t, and then the small t with the small t, and the capital T with the small t, and the small t with the capital T. So the resultant was like this, that there was a formation of TT, both were pure tall t, and this was the capital T small t, and this was the both were the short t. So this led to the construction of some plants. So the pure T, pure tall, pure capital T, they were tall. These were also tall, and this was short. But the one plant which was both genotypically both were tall capital T, this is called as a true or the pure tall plant, and these were called as a both. It contained the small t and also the short t, the tall t, and this is called the short t. Both were short t. So the tall t receive the 25% of the F1 generation genes, and the both phenotypically these were as tall, but genotypically it contain the gene of short plant. These were 50%, and the completely short plant were 25%. And the pure tall plant we call as a homozygous plant, while the both heterozygous, but while the tall plant which were genotypically also contain the genes of the short plant, these were called as a heterozygous. And the short plant, which was the pure short, this is called is a homozygous because it contained the genes of both genes of short plant. So if we look at the conclusion, so in this experiment, the traits which were masked in F1 generation reappeared. So you can see here the traits which were masked in the F1 generation. So in F1 generation, the trait of small t were masked. Small t were masked. You can see here, small t was masked. It was not appeared due to the dominance of the tall t. So the traits which were masked in the F1 generation, they were reappeared in the F2 generation. You can see here, this was masked, so it reappeared. You can see here. And approximately in F2 plants, one of the fourth recessive character and 3/4 were exhibit as dominant. So you can see here, 1 to 4th plant are small. 1 to 4 are small, while 3 to 4 are tall, because it is pure tall, but these two are genotypically not pure tall. So in the F2 generation, there was the formation of two type. The 1-4 was the short plant, while 3 to 4 was the formation of tall plant. So the ratio of dominance to the recessive was 3 to 1. It means that there was a formation of 3 tall plant while 1 short plant. So in the F2 generation, in one self-pollination, 3 plants were tall, 3 were tall, while the 4th one was short. In every self-pollination of F2 generation, 3 were tall, 1 was genotypically pure tall, 2 were genotypically not, 2 were not pure tall, and the 1 was pure short. So this was the law of segregation of Mendel, in which Mendel concluded that each individual contains some pictures per every character of a body, which during the gamete formation separate from each other. So therefore they separated from each other and form the short plant, the pure short, pure tall, and these were also tall but not genotypically. So therefore there was a formation of pure tall plant. So therefore Mendel concluded that each individual contains some factor per every characters which separate during the gamete formation, and they goes one to each gametes, not both to one gametes. So I hope that this lecture maybe will be very helpful to you, and you'll mix in next lectures.